Good evening students. So yes, in our previous class we have done till the kingdom Monera from biological classification, right. So once let us recall the fundamental insights of this chapter. So far we have dealt with kingdom systems of classification, kingdom Monera, and now we are going to begin with kingdom Protista. Today we will complete kingdom Protista and kingdom Fungi up till classification level, and then we will exercise few objective, okay, questionnaire round will be there, where we will practice NEET previous year questions also. So listen attentively, okay, your preparation will complete here itself if you are listening attentively. So first we will look into the general characteristics of the kingdom Protista. Boundaries of this kingdom are not well defined. What does this mean? Boundaries of this kingdom are not well defined, which means this forms a link with other kingdoms as well, because kingdom Protista includes what? All the unicellular eukaryotes. Okay. What does kingdom Protista include, beta? All the unicellular eukaryotes, unicellular eukaryotic organisms. Fine. These unicellular eukaryotes can have cell wall over them, which can resemble plant like protists, and few of them can act as decomposers, which will resemble like fungi like slime molds, and few of them will not have cell wall and their pure mode of nutrition will be heterotrophic, which are animal like protists called as protozoans. So this forms a connecting link between prokaryotes and eukaryotes. Okay, kingdom Protista forms a connecting link between prokaryotes and eukaryotes. Fine. And this includes all the unicellular eukaryotes. As they resemble plant like, fungi like, animal like protists, the boundaries of this kingdom are not well defined. Till here it is clear, right? Few of the general characteristics. Okay, you can expect statement based questions from this. So that is why I am stressing you out the all statements that are in reference with Protista. Next, general characteristics of the kingdom Protista. Here you see, they include what? As we have discussed, single celled eukaryotes, and they are primarily aquatic. Remember this term, they are what? Primarily aquatic. Once statement based asked questions, Protistans are primarily aquatic organisms. They have membrane bound organelles because they are eukaryotes, and they have well defined nucleus. Ribosomes will be of 80s, and flagella and cilia, both motile structures will be there. In Monera, if you have to consider, the only motile structure it has is flagella, but here you have both flagella and cilia, and the microtubular arrangement is 9 plus 2. In Monerans you have 9 plus 0 microtubular arrangement. And what protein is composed in the flagella? Tubulin protein. Whereas in the kingdom Monera, for flagella, flagella is composed of, very good, flagellin protein. Here it is tubulin protein. Clear till here? No? Any doubt anywhere? Yes. Next, nutrition. It exhibits all the modes of nutrition because it includes plant like, fungi like, animal like protists. Holophytic, plant like protists like chrysophytes and dinoflagellates are purely conducting holophytic mode of nutrition. Holozoic, animal like protists, those are protozoans, will conduct holozoic mode of nutrition. Holophytic, plant like protists, plant like protists. Fine. And mixotrophic mode of nutrition is conducted by euglenoids. In the presence of sunlight, it has photosynthetic pigments, that is chlorophyll A and B. It will perceive the sunlight and it will conduct the photosynthesis. In the absence of sunlight, it conducts the mode of saprophytic nutrition. Okay, so it includes both autotrophic and heterotrophic nutrition. That is why we say mixotrophic mode of nutrition. Saprophytic mode of nutrition is by slime molds, and protozoans will conduct parasitic mode of nutrition. Okay. So this kingdom has vast diversity in mode of nutrition. Understood, right? Next, reproduction. Reproduction can be both asexual and sexual mode of reproduction. Asexual mode of reproduction will be by fission and spore formation. Sexual mode of reproduction will be by cell fusion and zygote formation. Till here it is clear, right, beta? Next. Now we will look into the classification of Protista. As we have discussed, first is what? Photosynthetic or plant-like, which has what? Cell wall, okay, and it conducts autotrophic mode of nutrition except euglenoids, which has mixotrophic mode of nutrition. This includes what, Nana? Chrysophytes, dinoflagellates and euglena. Here you see consumer decomposer type of protists which are fungi like protists, include slime molds. In slime molds we are going to learn again about cellular slime molds and acellular slime molds. Here you see animal like protists or protozoans, which include amoeboid protozoans, flagellated protozoans, ciliated protozoans and non-motile sporozoans. Clear, right? You have got a basic idea on the framework of classification. Yes. Next, photosynthetic protists. Photosynthetic protists include what? Protista and algae. They are also called as what? Protista and algae, because they conduct autotrophic mode of nutrition. Okay. Major portion of them are acting as what? Phytoplankton. Okay. Major portion of them are phytoplankton and they are chief producers of oceans. Okay. Photosynthetic protists are chief producers in the oceans. First is chrysophytes. Chrysophytes includes diatoms and desmites. Desmites are also called as golden algae. Okay. Diatoms and desmites are also called as golden algae. Mostly they are aquatic, freshwater or marine organisms. Okay. They are both in freshwater and marine water habitats, you can see. Fine, right, ma? Next. So, they are microscopic and they float passively on the water currents. Okay. As I told you, most of them, major portion of them act as what? Phytoplanktons. They are the chief producers of the oceans. Remember, many at times asked questions for diatoms and desmites. And nutrition is holophytic, we have discussed, and photosynthetic pigments include chlorophyll A, chlorophyll C, xanthophylls and carotenoids. Fine. Diatom cell wall is made up of indestructible silica. Okay, it has what? Siliceous cell wall. Diatom cell wall is siliceous. It has siliceous cell wall. Okay, diatom has two thin overlappings. The upper overlapping is called as epitheca, lower one is called as hypotheca, which resembles a soap box like structure. Both epitheca and hypotheca together you will call it as frustule. Okay. So, upper one is called as what, Nana? Epitheca, lower one is called as hypotheca, both together you will call it as what? Frustule. Clear, right? Next. Here we go. As the cell wall is composed of silica, it has siliceous cell wall. The diatoms, when they fall on the seabed, okay, they form diatomaceous earth, which is also called as Kesselgar. Okay, this is having many, having much of commercial importance. Like this is used in polishing, filtration of oils and syrups. Okay, that is the commercial importance of what? Diatomaceous earth or Kesselgar. It has cell wall. Cell wall is composed of what? Diatom cell wall is composed of what? Silicon or silica, siliceous cell wall you will say. Okay, when they fall on the seabed, when they die, that siliceous cell wall will get deposited over the seabed. Okay, that depositions are called as what? Diatomaceous earth or Kesselgar, which has high amount of commercial importance, and this is the mode which you use in the commercial perspective. Next, based on the symmetry. Okay, based on the symmetry, diatoms are of two types beta. They are centrales and pinnales. Centrales will show radial symmetry, that is, we can divide the body in any plane, any plane the body is divided into two equal halves. So, centrales type is having radial symmetry and pinnales is showing bilateral symmetry, that means by only one plane you can divide the body into two equal parts. Okay. So, clear with this centrales and pinnales? Any doubt anywhere, ma? Next, reproduction. We have both here in diatoms, asexual and sexual reproduction is there. First, asexual mode of reproduction, most common mode of reproduction, that is binary fission, which occurs in favorable conditions. Sexual reproduction is through gamete fusion. Okay, and after gamete fusion, the zygotic structures which are formed are called as oxospores. These oxospores are known as rejuvenatory spores, because again when the favorable conditions are resumed back, they will regain their shape and structure. Okay. So, why oxospores are called as rejuvenatory spores, ma? Okay. They resume the shape and size. They resume the shape and size back when the favorable conditions are back. In favorable conditions you can take. Okay, whenever favorable conditions are back, they will get back into their normal shape and size. That is why oxospores are called as what? Rejuvenatory spores. So, first class of plant like protists is clear to you know, chrysophytes, okay, which includes what, no, diatoms and desmites, which is golden algae. Clear, no, about pigmentation, about reproduction, about nutrition, okay, and about its commercial importance, everything is clear, right? Now, dinoflagellates. Dinoflagellates are also called as golden brown photosynthetic protists. Don't worry, I will send this PDF to your group, okay. So, they are also called as what, ma? Golden brown photosynthetic protists. They are mostly marine, okay, unlike diatoms which are both freshwater and marine habitat. Dinoflagellates are mostly marine. They include photosynthetic pigments like again chlorophyll A, C, beta carotene and xanthophylls. The pigmentation is same in both chrysophytes and dinoflagellates, okay. And here when we see the cell wall composition, cell wall of chrysophytes is made up of what, ma? Silica, siliceous cell wall. But here the cell wall is made up of stiff cellulosic plates. Diatom cell wall is made up of stiff cellulosic plates. Nucleus in diatoms is called as, is noted as mesokaryon, okay, which resembles both the properties of nucleus in prokaryotes and a nucleus in eukaryotes. Nucleus in eukaryotes because the nucleus is having nuclear membrane. As it is having nuclear membrane, this is resembling with that of the eukaryotes. In prokaryotes, how is it resembling with prokaryotes is, at the time of nuclear division, that is karyokinesis, there is no involvement of spindle apparatus formation. It is like amitosis only, okay. No spindle apparatus formation at the time of karyokinesis. Clear, right, ma? Yeah. So, nucleus is called as mesokaryon because it shows properties in resemblance with both prokaryotes and eukaryotes. Eukaryotes because the nucleus is surrounded by the nuclear membrane, prokaryotes because there is no spindle apparatus formation. Clear till here, right? Next is flagella. Flagella, we have two types of flagella, one is longitudinal and second is transverse. Transverse flagella and longitudinal flagella. Both transverse flagella and longitudinal flagella will show whirling whip movement. That is a characteristic feature of dinoflagellates, many a times asked question in your NEET exam. So, flagella is having what? One longitudinal flagella and one transverse flagella which will show what movement with a whirling whip movements. This is characteristic feature of what? Dinoflagellates. Till here it is clear, right? Any doubt anywhere? Yeah. Next, bioluminescence. Marine dinoflagellates. Bioluminescence is the feature which is shown by marine dinoflagellates. Example you have to remember, that is Noctiluca. Now comes the concept of red dinoflagellates, okay, which causes red tides in the Red Sea. Okay. The same red tides in the Red Sea are observed by some of the cyanobacteria also, like Trichodesmium erythraeum, that we have learnt when we are learning the concept of cyanobacteria under the kingdom Monera. Okay. Do not get confused between these two examples. One is Trichodesmium erythraeum of cyanobacteria and the second one is Goniolax of the kingdom Protista. Okay. Both of them will show the same feature, that is causing red tides in the Red Sea. That is because of the toxins they release. Here for Goniolax though, because of their rapid division, they release the toxins. Those toxins are called as saxitoxin. This will kill the marine fishes and this causes red tides in the Mediterranean Sea. Till here it is clear, right? Whereas there in the cyanobacteria, what is the example I gave you? Trichodesmium erythraeum. Same phenomena you will see, that is causing red tides in the Red Sea. But you should be clear while answering based on the question. whether he is asking you to identify a cyanobacterian type which is causing red tides in the Red Sea, or the question is about dinoflagellates which causes red tides in the Red Sea. Clear with this too, right? Yeah. Next. No, some other, like you remember only this term ki red tides are caused with both cyanobacteria and dinoflagellates. Be clear on the example. Clear, right, Raghav? Okay. Yeah. Next is Euglena. Euglena belongs to what? Euglenoids. Okay, plant like protist. But then here cell wall is not composed of silica like that you see in the diatoms, or cellulosic plates like that you see in the dinoflagellates. Euglena, cell wall is covered, there is no cell wall, a proteinaceous layer is present, that is called as pellicle, which gives flexibility for it during movement. Okay. Is there any cell wall here? Cell wall is absent, but protein rich layer pellicle is present. Okay. Mostly these are what? Fresh water. Dinoflagellates are mostly marine water, and chrysophytes are both fresh water and marine. Remember about habitats as well. Now you look into the structure of Euglena. This region is called as paraflagellar body. Okay, this region is called as what? Paraflagellar body. Yeah, this is paraflagellar body. From paraflagellar body, one longitudinal flagella will arise and one short flagella. Longitudinal flagella will have hair-like structures called as stichonemes. Okay, this is called as tinsel flagella, or both together stichonematic flagella, and there is a pigment, photosynthetic pigment attached to the paraflagellar body. That photosynthetic pigment is called as astaxanthin or eye spot. That is called as astaxanthin or eye spot, and short flagella and long flagella. This is clear to you know. And then contractile vacuole, which helps in osmoregulation. Nucleus is membrane bound and protein layer, that is pellicle. Clear about the structural description of Euglena? Any doubt anywhere, Vita? Yeah. So, mixotrophic mode of nutrition. Photosynthetic, it is photosynthetic in the presence of sunlight, which shows holophytic mode of nutrition, and the photosynthetic pigments are same that you see in the higher plants. If you look into the previous two, that is dinoflagellates and chrysophytes, you have observed chlorophyll A and C. Fine, but here you can see chlorophyll A and B. These are the photosynthetic pigments. Chlorophyll A, B, xanthophylls and beta carotene. These are the photosynthetic pigments. Okay. Next, heterotrophic mode of nutrition. Heterotrophic mode of nutrition or holozoic mode of nutrition. This occurs in the absence of sunlight, wherein the mode of nutrition is saprophytic. This gets nourishment from dead and decaying organic matter. Okay, clear till here? Any doubt anywhere? Yeah. Next, reproduction. There is no sexual reproduction for euglenoids. Okay, there is only one mode of asexual reproduction, that is longitudinal binary fission. Okay, longitudinal binary fission, and during unfavorable conditions they go through a palmella stage. Okay, palmella stage where cysts are formed. Cysts will act as organs of perennation. Whenever favorable conditions are resumed back, from cysts euglena will arise and they will conduct the longitudinal binary fission. Clear? Raghav, I can see some question mark. Clear, right? Yeah. Next is fungi like protists. Fungi like protists include what? Slime molds. Okay, slime molds, how can you describe them as with a achlorophyllous saprophytic protists. Okay, and their locomotion is through the false feet called as pseudopodia. Okay, pseudopodia will move like that. Okay, the movement of pseudopodia is this, which is false feet. Okay, called as false feet. Pseudopodia you will also see in the amoeboid protozoans. Okay, and what is the vegetative stage of this? That is multinucleated mass of protoplasm, which is surrounded by a plasma membrane. Cell wall is absent here. Okay, cell wall is absent in the vegetative stage of the slime molds. Remember, but cell wall is present in the spores that are released from the slime molds plasmodium during unfavorable condition. Remember the statement properly, ma. Okay, the vegetative stage is multinucleated, surrounded by cell membrane, it does not have any cell wall. Okay, and during the favorable conditions, slime molds will extend their feet. Okay, and they will form aggregations called as plasmodium. Okay, during favorable conditions, what happens? Slime molds will extend their feet and they will form what? Plasmodium. That aggregations are called as plasmodium. During unfavorable conditions, from plasmodium only fruiting bodies will arise, and at the tip of fruiting bodies spores will be released. Okay? Those spores are having cell walls. Vegetative stage will not have cell wall, but spores will be having the cell walls. These spores are released into the air currents. Okay? Spores are having what? Cellulosic cell wall. Till here it is clear now? Now we will see the clear differentiation between cellular slime molds and acellular slime molds. Cellular slime molds habitat occurs on humus containing upper layer of damp soil. Okay? And somatic phase you will see formation of pseudoplasmodium also you will observe. Just you have to remember cellular slime molds, you will have uninucleated condition for each cell. Acellular slime molds, cenocytic condition is observed in the cells. And what you have to remember is the examples. Dictyostelium and polyspondylum are the two examples under cellular slime molds. Fusarium and Fusarella are the examples of acellular slime molds. Clear with slime molds now? Any doubt anywhere ma? No, right? Yeah. Now the third division under the kingdom Protista, those are animal like protists which are protozoans. So protozoans, if you see, these are the primitive relatives of the animals. Okay? Heterotrophs, and they act both as predators and parasites. Cell wall is completely absent. We have four divisions: amoeboid, flagellated, ciliated and sporozoans. Let us learn all together. Amoeboid protozoans are having false feet called as pseudopodia, and their cell wall is made up of silica again, and it is surrounded by silica, siliceous cell wall, that is a characteristic feature of it. Just a second. Siliceous shells, not cell wall. It has siliceous shells. Next is paramecium. Paramecium will have cilia around it. The body is surrounded by the cilia. Ciliated protozoans there. They have binucleated condition, macronucleus and micronucleus. Macronucleus is involved in the conduction of metabolic activities of the paramecium. Micronucleus will go through the reproductive activities of the paramecium. Okay? Macronucleus for nutrition, micronucleus for reproduction. Fine. Next is flagellated protozoans. Example is trypanosoma. If at all there is a question like whether flagellated protozoans or ciliated protozoans will show fastest movement, which of these two will show fastest movement? If this is the question, your answer should be ciliated protozoans. Ciliated protozoans will show fastest movement when compared to flagellated protozoans. Clear, right? Next is non-motile sporozoans. Example is plasmodium. Now we will learn all together in a tabulated form. Okay. First, amoeboid protozoans, habitat if you see ma, fresh water, marine water and moist soil. This is the habitat. Next habit is free living, and few will act as parasites, and then locomotion is through pseudopodia. Distinct features, they are marine forms, like they have what? Silica shells. Examples include amoeba and entamoeba. Next is flagellated protozoans. They are free living aquatic protozoans. Their habitat parasites, they act as parasites. Locomotion is through flagella. Example is what? Trypanosoma. Examples are very important, and key features, distinct features of each class is important. Ciliated protozoans, aquatic, few act as freshwater, few are parasites amongst them. Cilia is their motile structure, and they have cavity to engulf the food to transport, that is gullet. Okay. Example is what? Paramecium, and you can also take under the key features that it shows binucleated condition, macronucleus and micronucleus. Next is sporozoans. Sporozoans are endoparasites. They show parasitic habitat, no cell wall, no motile structures. They form an infectious stage like spore like stage. Okay, you will see in the malaria life cycle. Okay, and the example includes what? Plasmodium. Clear with all the types of protozoans, right? Clear everywhere? Yeah. Now we will move to fungi. Kingdom fungi, first let us focus on the general characters. Fungi, this is a unique kingdom where it is eukaryotic and thalloid, multicellular, loose tissue organization you will observe. Okay? Eukaryotic, thalloid, achlorophyllous, heterotrophic organisms, purely heterotrophic. They show great diversity in morphology and habitat, but not in the mode of nutrition, because mode of nutrition is purely heterotrophic. Clear, right? Now, they vary in their morphology from unicellular yeast to complex edible mushrooms or toadstools. They vary from unicellular yeast, Saccharomyces cerevisiae, to complex edible mushrooms or toadstools. Fungi is cosmopolitan in its distribution. Mostly it occurs in warm and humid places. This is the habitat of it. Clear with the general characters? Let us continue with few more. Fungi will have slender thread-like structures. Those are called as hyphema. The network of the hyphae is called as what? Mycelium. Mycelium is again of two types, if at all septa is present amongst them or not. In few primitive fungi like Phycomycetes, septa will not be present. So, that shows what? Aseptate condition. In aseptate condition, the long tubes of hyphae are filled with what? Nucleus, which is multinucleated or cenocytic condition. So, aseptate mycelium will show cenocytic hyphae, which forms continuous tubes, for example, Phycomycetes. Whereas, septate mycelium which has septa in between, right? Each septa will have one nucleus, okay? Uninucleated condition, septate or cross walls are present. Includes Actinomycetes which has pore-like septa, Basidiomycetes which has dolipore septa. Pore-like septa, where septa is through pores like this, fine. Dolipore septa is like this. Dolipore septa is the characteristic feature of the advanced class of fungi, that is Basidiomycetes. And lastly, Deuteromycetes is also showing septate mycelium. Clear with hyphae? Okay. Clear with mycelium? Okay. Network of hyphae? You are clear with aseptate mycelium and septate? Excuse me. Aseptate is showing cenocytic condition. Septate is showing septa. Pore-like septa you will observe in Actinomycetes. Dolipore septa you will observe in Basidiomycetes. Cell wall. Cell wall is composed of chitin and polysaccharides. This chitin and polysaccharide composition you will also call it as what? Fungal cellulose. Chitin is also called as fungal cellulose. Nutrition, as we have discussed, it is purely heterotrophic. Nutrition is purely heterotrophic. Fine. First mode, if you see, saprophytic mode of nutrition. How saprophytic mode of nutrition is conducted? The mycelium body will release some digestive enzymes into the surroundings where the food is in the close proximity. These enzymes will degrade the complex substance into the simpler substance. Complex substance will be degraded into simpler substances, and these simpler substances will be absorbed by the mycelium. This is about saprophytic mode of nutrition. Parasitic, like albugo, which causes leaf infections in the mustard leaves. Okay, parasitic mode of nutrition. And symbiotic mode of nutrition, best and first example is lichens. Symbiotic mode of nutrition is lichens. Just a second. Yeah, fine. So, symbiotic mode of nutrition is what? Symbiotic mode of nutrition example is lichens, that is fungal association with algae. Second is fungal association with the roots of higher plants like pinus. Here the roots of pinus are called as mycorrhizal roots. Mycorrhizal roots, because it has fungal associations. Okay. Next, reserve food material is glycogen and oil. This is the reserve food material. You are clear with mode of nutrition, you are clear with cell wall, you are clear with mycelium, right. Next is reproduction. Reproduction can be done through three modes of reproduction in fungi. First is vegetative mode of reproduction is through fragmentation, fission and budding. Okay. Next, asexual mode of reproduction is through spores. Both asexual mode of reproduction and sexual mode of reproduction is through spores. Asexual spores and sexual spores. Asexual spores and sexual spores. So, here asexual spores include conidia, sporangiospores and zoospores. Sexual spores include what? Oospores, ascospores and basidiospores. Clear, right? So, here we will look into each mode of reproduction in detail. You have to remember the asexual spores and sexual spores, very important. Sexual spores, oospores you will see in oomycetes or phycomycetes, ascospores in ascomycetes, basidiospores in basidiomycetes. Vegetative reproduction, here we go. Fragmentation, where the mycelial hyphae will be fragmented into small hyphae and this hyphal structures will give rise to the entire mycelium. This is fragmentation mode, you all know it clearly. Next is budding. Budding, where from the parent bud daughter buds will arise. This daughter buds landing onto the suitable substratum will generate a new hyphae. That is about budding. Next, binary fission, that is the most common mode of reproduction in unicellular fungi like yeast. 3 modes of reproductions are clear, right? Under vegetative reproduction. Next, we will go through asexual and sexual reproduction through the life cycle first. Okay. So, as I told you, both asexual and sexual mode of reproduction is through what? Spores, right. So, here from the mycelium post mitosis asexual spores are released. What are the asexual spores? Conidia, sporangiospores, zoospores, aplanospores. These are the asexual spores. These spores landing onto the suitable substratum, they will again give rise to mycelium. Remember, fungal body is haploid. Fungal mycelium is what? Haploid. So, after mitosis it will produce spores which are haploid. Okay. Haploid spores landing onto the suitable substratum, they will again give rise to haploid mycelium. Clear with the asexual cycle beta? Now, sexual cycle. Sexual mode of reproduction is operated in ascomycetes, basidiomycetes and phycomycetes. In phycomycetes, though, it is primitive mode of sexual reproduction, whereas ascomycetes, basidiomycetes, sexual reproduction takes place in 3 stages. Okay. Sexual reproduction occurs in 3 stages. First is plasmogamy. Second will be karyogamy. And third will be meiosis. Okay, plasmogamy, karyogamy and meiosis. Plasmogamy, where the cytoplasm of 2 different strains of mycelium will be exchanged first. Okay, that is plasmogamy. Look into the description also. Haploid cells from 2 different mycelial strains will fuse, forming a heterokaryotic cell. This heterokaryotic cell post plasmogamy, you can see N plus N condition. Karyogamy is what? Nuclear fusion. The nucleus, that is N plus N condition, this will fuse and they will form a diploid nucleus. They will form what? Diploid nucleus. And this forms out to be what? Zygote. Okay. Zygote post meiosis. Okay. Zygote is diploid one, right. So, after post fusion, this is forming a diploid nucleus to diploid zygote. This diploid zygote will undergo meiosis. What type of meiosis? Zygotic meiosis. Like how we have seen zygotic meiosis in algae, same ways here also it is zygotic meiosis. After zygotic meiosis, haploid spores will be released into the suitable substratum, then it will give rise to haploid mycelium. Clear with sexual cycle and asexual cycle? Any doubt anywhere? I will give you 1 minute of time, just go through. Okay, if you are having any doubt, let me know. Good, Bharat. At the end, you please stay for the questionnaire session also, ma, where you can practice it. Okay. Clear? Here we go. Asexual reproduction, as I told you, with asexual spores. Zoospores are motile and biflagellate. Conidia are non-motile and they are exogenous, okay, and they are released from the tips of conidiophores. Sporangiospores, they are thin walled, okay, and they are non-motile. They are released from the bodies of sporangiophores. They are endogenously released. Here you remember one thing, ma, the only asexual spores which is released exogenously is conidia, okay, and there is one more spore to be highlighted under asexual spore, that is aplanospores. Aplanospores, which are non-motile spores. And during unfavorable conditions, one more type of spores are released. Those are called as chlamydiospores. Chlamydospores, which are released in unfavorable conditions. Clear, right? Zoospores, which are motile and biflagellate. Aplanospores are non-motile. Conidia, they are non-motile and exogenously released, where spores are released outside, and then from the tips of conidiophores. Sporangiospores, they are thin walled and they are endogenously released. One point to be remembered, apart from all this, only exogenously released asexual spore is what? Conidia. Then about sexual reproduction. Sexual reproduction is through what? Sexual spores. Sexual spores like oospores, ascospores, basidiospores. Okay. Oospores are non-motile and endogenously released, observed in Phycomycetes. Ascospores in the class Ascomycetes, wherein these are non-motile endogenously produced from ascocarps. Ascocarps are fruiting bodies over here. And then basidiospores, these are non-motile exogenously released spores from the fruiting bodies basidiocarps. Clear, right? You have to be clear with the types of spores and the classes in which they are mentioned. Ascospores are from Ascomycetes, basidiospores, Basidiomycetes. Okay? And only exogenously released sexual spores are what? Basidia or basidiospores. Clear, right? Any doubt anywhere? Yeah. Important slide, ma. Many at times the questions are put only on the sexual and asexual reproduction. That important it is. Okay. Next. Based on the morphology. Now we will look into the classification and then we will go with the questionnaire practice. On what basis the classification of fungi is done? On what basis? On the basis of morphology of mycelium, mode of spore formation and fruiting bodies. Okay, on the basis of morphology, mode of spore formation and fruiting bodies. On this basis, classification of fungi is done. One time asked question in your NEET PYQ, that fungal classification, is it based on mode of nutrition? Mode of nutrition? No. Why? Yes, only mode of nutrition is heterotrophic mode of nutrition. That is why for classification of fungi, mode of nutrition is not the criteria. What are the three criteria, ma? Morphology of mycelium, mode of spore formation and fruiting bodies. Here we go with the classes of fungi. Phycomycetes again includes two subclasses, Oomycetes and Zygomycetes. Phycomycetes is also called as algal fungi and this is the primitive fungi. Okay? Phycomycetes, algal fungi or primitive fungi. Okay? Ascomycetes known as sac-like fungi or sac fungi because it has sac-like fruiting bodies called as ascocarps. Next, advanced group of fungi, that is Basidiomycetes. Basidiomycetes are known as puffballs or bracket fungi. The most advanced class of fungi because its only mode of reproduction is sexual mode of reproduction and it has dolichopore septa. Asexual mode of reproduction is absent in Basidiomycetes. Deuteromycetes is called as fungi imperfecti or imperfect fungi because it does not have any sexual stages in its life cycle. Okay? This is called as what, ma? Fungi imperfecti. Clear? Any doubt anywhere? Yeah. Next. Questionnaire session. Okay? These questions I have put both from Protista and till the classification of fungi. Look into it carefully and answer. Correct statement need to be chosen. Online students, even you can answer, ma. Statement. If statements, I will give options, right, ma? 1 and 4 बोल के. Very good. Why first option is wrong? Cholera, typhoid, tetanus are well known diseases caused by bacteria, not by viruses. Okay. Cholera is by diphtheria, cholerae. Typhoid, salmonella typhi. Tetanus, forgot, right? Very good. I have given no, right? Harmful bacteria. Revise them again. Dinoflagellates, euglenoids and slime molds are placed under the kingdom Protista, not Monera. Members of the kingdom Protista are primarily aquatic. True. Dinoflagellates are the chief producers of the ocean. That is why I have mentioned you clearly. What are the chief producers of the ocean? Chrysophytes, that is diatoms, not dinoflagellates. You only go back and see in your class notes, no? No, open your NCRT right now, right away. I'll show you after the questionnaire practice, okay? Yeah, but the right statement is members of the kingdom Protista are primarily aquatic. That habitats also I have stressed out. Look at here. Select the incorrect statement. Carefully observe and answer. Very good. How 3, Abdul? It's 4, Nare. The cell wall of mycoplasma are made up of chitin. Incorrect, because mycoplasma will not have any cell wall. Okay. Next, this question. So, the right answer here is option 4. Yeah, this one. Non-Protestian group. Very good. Belongs to the kingdom fungi. Belongs to the kingdom Raghav? Yeah. This one. Which of the following is correct? Slime molds are haploid, protozoans lack cell wall is correct. Yes, slime molds are haploid, but the spores are diploid. Dinoflagellates are non-motile, wrong. Pellicle is absent in euglena, wrong. So, the direct straightforward answer is what? Protozoans lack cell wall. Okay, this one. Which is not a feature of dinoflagellates? Very clear and evident statement. Clear? Which option? 4th, because they are photosynthetic, not non-photosynthetic. Next. This one you see, ma. Slime molds are saprophytic protists. Good, Raghav. This one. In 5 kingdom classification, which of the following groups of organisms is placed in the same kingdom? If you answer this, na, see. Good. %. Answer carefully, Ray. C. Chlorella belongs to algae. We can also consider it as Protista because it is unicellular. Amoeba, Protista. Paramecium, Protista. Spirogyra, algae. Okay. Chlamydomonas, amoeba, goniolax, chlorella. Again, few of them are algae, few of them are Protista. Spirogyra, nostoc and Anabaena belongs to kingdom Monera because both of them are bacteria. So, this you cannot consider. Spirogyra, Chlamydomonas, Euglena, nostoc again is a BGA, right, which belongs to kingdom Monera. So, this also you cannot consider. Chlamydomonas is multicellular fungi, multicellular algae, filamentous algae, sorry, unicellular algae. I thought it is cladophora, I am sorry. Chlamydomonas, unicellular algae, amoeba, goniolax and chlorella. No, few of algae are there, few of protistans are there. Here we go with the first option, chlorella, amoeba, paramecium and spirogyra. All of them are unicellular. Few you can put into the plant like protist, few as fungi like, protozoans, okay, amoeba and paramecium, okay. Done. Option 1 is right over here. Good. Next. This one. The fungi of Euglena is flexible due to the presence of good. This one. NEET 2024. Good. Option 2, right? Yeah. Last but one question. Match list 1 and list 2. Sure, confident? We will see. Euglenoid, option 4, right? Dinoflagellate, photosynthetic. Slime molds, parasitic. And Plasmodium, Plasmodium parasitic. Wait. Euglenoid is option 4. Okay. Dinoflagellates we can say photosynthetic, option 3. Yes. Slime molds, saprophytic, right? Parasitic. Both we can include. Plasmodium. Plasmodium is parasitic, which is a protozoan. So, here C is, A is 4, B is 3, C is 2, D is 1, option 3, right? Yeah, next. You said option 3 only, na Abdul? Good. Last question. Answer carefully. NEET 2022 question. Sure? Okay, we will see. Correct statement need to be identified. Bacteria are exclusively heterotrophic. No, they are both autotrophic and heterotrophic. So, we can eliminate this. Slime molds are saprophytic organisms classified under the kingdom Monera. Wrong. They are classified under the kingdom Protista. Mycoplasma has DNA, ribosome and cell wall. Cell wall is absent in the mycoplasma because it is primitive bacteria. Next, cyanobacteria are a group of autotrophic organisms classified under the kingdom Monera. Option 4 will be the right answer. Clear? Any doubt anywhere? Till here everything is fine, right? Yeah. So, with this we have done till half of the kingdom fungi. Rest of the kingdom fungi we will learn each and every class of kingdom fungi in detail. Post that Plantae and Animalia in brief account. After that, those organisms which are not included in 5 kingdom system of classification like virus, viroids, prions, lichens. With that we can end with end this chapter. Okay, one more class is required. We can finish off this chapter and start plant kingdom. Okay, so that's all for today's session, beta. Thank you.